Nobuyuki Takakura, MD, PhD, from Research Institute for Microbial Disease, Osaka University, Japan. So, ladies and gentlemen, today's event is organizing by Master and Doctoral Program in Biomedical Sciences, Faculty of Medicine, Universitas Indonesia, in collaboration with Stem Cell and Tissue Engineering Research Center, Faculty of Medicine, Universitas Indonesia. So before we begin the session, there will be an opening remarks from the Secretary of Doctoral Program of Biomedical Sciences, Faculty of Medicine, Universitas Indonesia. Right now, present with us, Dr. Robiatul Adawiyah, Master of Biomedical Sciences, Specialist of Clinical Parasitology. Dr. Robiatul Adawiyah is here with us, Doctor?
Yes, I'm
Okay.
here. Thank
So
you.
without further ado, to Dr. Robiatul Adawiyah, the time is yours.
Thank you very much, Nuzli, for the introduction. Could you hear my voice clearly?
Yes, Doctor.
Okay, thank you. Okay, here. Assalamualaikum warahmatullahi wabarakatuh. Good evening, Professor Takakura, and also all the audience. Here I'm represent the head of doctoral program in biomedical sciences, Faculty of Medicine, Universitas Indonesia. This guest lecture with the title Angiogenesis and Vascular Endothelial Stem Cells will be delivered by Professor Nobuyuki Takakura from Department of Signal Transduction Research Institute for Microbial Diseases, Osaka University, Japan. Okay, thank you very much, Professor Takakura. I hope this guest lecture will encourage our students to explore more about stem cell and also our college here in a doctoral program of biomedical science, FMUI. And thank you very much for this collaboration. I hope this collaboration will be better in the future and could be more useful for all of us. I think that's all for me. Thank you. And times is your to MC, Miss Nuzli Fahdia. Okay, thank you.
Okay, thank you so much, Dr. Robiatul Adawiyah. Now, we welcome also the Secretary of the Master Program of Biomedical Sciences, Faculty of Medicine, Universitas Indonesia. Here with us, Dr. Rani Wardani Hakim, Master of Biomedical Sciences. Dr. Rani?
Okay, thank you, Ms. Nuzli.
Okay.
Good afternoon, everyone. Good afternoon, Professor Takakura. I'm here, Rani Wadani Hakim, on behalf of Head Magister Program in Biomedical Sciences, Professor Warren Muri Aroza. I extend welcome and thank you to Prof. Takakura for your willingness to give us lecture about angiogenesis and vascular endothelial stem cell. Actually, our study program has about 16 concentration, and one of the concentrations is stem cell, which lead by Dr. Radiana. I'm very happy to have your lecture this afternoon because I hope your knowledge and your sharing can give our students, especially in stem cell concentration, spirit and then knowledge to do the research with more broad knowledge and then with more willingness to find something new, something interesting. And also, I hope another student who maybe didn't know about stem cell can gain a new knowledge about angiogenesis and vascular endothelial in stem cell. So thank you very much, Prof, to join with our program study this afternoon. I hope our student can have gain as much as they want to know about stem cell and angiogenesis. Thank you very much again. Then I will back to the moderator, Ms. Nuzuli.
Okay, thank you so much, Dr. Rani, for the opening remarks. And now, before we begin the lecture session, I want to introduce our moderator for today's guest lecture. Present with us, Dr. Radiana Dewayani Antarianto, Master of Biomedical Sciences, PhD. And Dr. Nana will guide us today through the discussion and also facilitate us through the question and answer session in the end of the lecture session. So Dr. Radiana is an associate in Department of Histology, Faculty of Medicine, Universitas Indonesia. And she's also a laboratory manager in the Stem Cell and Tissue Engineering Research Center, IMERI. And her research is focusing on iPSC cells, natural killer cell, and also the development of extracellular vesicle from the stem cell and other cells. And Dr. Adiana has several publications and awards, as you can see in here. The latest publication of Dr. Adiana is about the liver organoid and also the fibrosis in bile duct ligation model in the rabbit. For Dr. Adiana, present with us today, Doctor? Okay.
Yes, Leslie, thank you.
So everyone, let's all give a warm welcome to our moderator today, Dr. Adiana Dewayani Antarianto. Thank you.
Okay, thank you so much, Leslie, for the kind introduction. So, everyone, I'd like you all to meet a very excellent mentor and also a professor who I have the privilege to be in his lab, even if it was only for 3 months. But it has been eye-opening experience and to be able to do the research which to investigate the formation of the blood vessels inside the liver organoids. So I think I would like to first introduce our distinguished speaker for today. So Professor Nobuyuki Takakura obtained his medical doctor in Mi University Faculty of Medicine in 1988 and obtained his PhD from Kyoto University Graduate School of Medicine. It is spectacular to see the research and career history of Prof. Takakura, since he has subsequently been appointed 3 times as a professor. In 2001, he's a professor in the Department of Stem Cell Biology, Cancer Research Institute, Kanazawa University. In 2006, he is back again appointed as a professor in Department of Signal Transduction, Research Institute for Microbial Disease, Osaka University, until today, currently. And from 2018, he's also appointed as professor in Immunology, Frontier Research Center, Osaka University. And his list of publication is very meaningful. You can say Nature Communication as one of his avenue for publications, and then Cell Reports, Cancer Research, Developmental Science, Oncogenes, Diabetes. So all the high impact publication has been presented by Professor Nobuyuki Takakura and his team. So without further ado, I would like to introduce you to Prof. Nobuyuki Takakura Sensei. Prof, the time is yours. Please enlighten us with the science and technology of angiogenesis and vascular endothelial stem cells. Thank you, Prof.
Okay. Okay, so may I start anyway? Okay. Do you hear me? Okay.
Crystal clear, Prof. Thank you.
Okay, so thank you very much for your kind introduction. And also, first of all, I'd like to express my sincere thanks to all members of your program. And very happy to have this kind of the opportunity to talk my research in terms of the angiogenesis and also the vascular endothelial stem cell, which we recently identified. So anyway, before my talk, so already Radiana-san told So I would like to say thanks again, Radiana-san, for your contribution to our research. She stayed our laboratory from July 2024 to October 2024. So this was the picture taken at the time. So she is here, and I'm here. And we have another researcher from Indonesia, your university. So she's Faya-san, now doing that lysosomal diseases. And also the other is Fitoriana-san. She's doing that cancer cachexia associated with blood vessel formation. So anyway, I'd like to say thanks to Radiana-san for your giving me this opportunity to talk. Anyway, so before my talk again, I would like to introduce you our institute. So our institute is Research Institute for Microbial Diseases. So this is the abbreviation RIMD. So this is the surface and appearance of our building. But we have another, three buildings. This is one of the building, but we have another, 1, 2, 3, 4 buildings, our, research institute, Osaka University. And also, you don't know about that Osaka University so well, but Osaka University is, of course, located in Osaka, but we have 4 different campuses. One is the Nakanoshima campus, as you can see here. So this is located in the center of the Osaka riverside. And also we have Toyonaka campus and Mino campus and the Suita campus. So these campuses are very close. But anyway, we are in this Suita campus. So in this Suita campus, we have a department of medicine and pharmacy and dentistry, and also many medical research centers, including our institute. So the history of our institute, our institute was, established in 1934, so 90 years ago. So we initially focus on the infectious diseases. So we developed many, vaccines for many, virus, for example, the measles, chickenpox, and flu, malaria, and so on. And also after vaccine development, we also focus on the host defense. So immunology was, focused. And also in the 1970s, viral induced cancer and oncogenes was discovered from virus. So we focus on the oncology at that time. And then we also recently focus on the aging. So I'm, belong to oncology department in this institute. So today, I'd like to talk about the endothelial stem cell population. Anyway, by 2 topics. One is the isolation of endothelial stem cells in the pre-existing vessels. The other developments and the aging of endothelial stem cell population. So from the first topics. So blood vessels show very different structure in depending on organ. So liver contains sinusoidal blood vessels, very fine capillary formation, and the skeletal muscles have a spiral shape, and lung, especially alveolar, so balloon-like structure of the capillary is observed, and nerve neurovascular wiring is observed. So this structure is very different depending on the tissue and organ. But recently it has been reported that the gene expression of endothelial cells is also very different depending on the tissue by using the single-cell RNA sequence analysis. However, if you look at only one organ, lung has arterial endothelial cells, venous endothelial cells, and the general capillary endothelial cells, so-called GCAP, and also lung has alveolar capillary A-cap endothelial cells. So this is specifically called aerocyte. So this, is very important for O2 CO2 gas exchange in alveolar. So this is one of the heterogeneity of endothelial cells in lung, but also during angiogenesis. So this angiogenesis is a new blood vessel formation from the pre-existing blood vessels. So during this angiogenesis, there are three types of different endothelial cells. One is chip cells. So this is the this decides the migration direction of new blood vessels. And then behind these, chip cells, so always stalk cells adhere to chip cells, and they have ability to generate a lot of endothelial cells for elongation of new blood vessels. And finally, during angiogenesis, so these phalanx cells emerge for the stability of blood vessels. And this angiogenesis is closely associating with the, tissue regeneration, tissue development. Therefore, these endothelial cells produce the tissue-specific angiocrine signals such as HGF, Wnt2 in the liver, Nogin in the bone marrow for osteocyte, and S1P in the pancreas. So this angiogenesis, there are three types of different endothelial cells emerge. So, endothelial cells show the phenotypically and functionally very diversity. So based on this diversity, we thought that endothelial stem cell populations may exist in the pre-existing blood vessels. So, the organ-specific stem cell population have been already, discovered. For example, the hematopoietic stem cells in the bone marrow, and intestinal stem cell population in the crypt, and also the neuronal stem cell population in the hippocampus, in the brain, and the skin stem cell population in the biology of the dermis. So, however, the endothelial stem cell population has not been identified so far. So if you, imagine, if the endothelial stem cell population exists in the pre-existing vessels, so we can very easily imagine the integrity turnover of vessels and also the aging. So in terms of the integrity, so stem cell population exists, they have ability to self-renew, and one of these endothelial stem cell like progenitor cells, they produce a lot of endothelial cells for the sustainable blood vessel formation. And also in the case of the aging, so in the aging, I will show later, but in the aging, decrease of capillary density is induced. So probably this decrease of capillary density is caused by the decrease of the endothelial stem cell population in the aging. So this we can easily imagine how the decrease of capillary density is induced in aging. So anyway, in order to isolate the endothelial stem cell population in pre-existing blood vessels, we firstly utilize the DNA dihexist method. So usually stem cell population express abundant ABC transporter pumps, Therefore, once stem cells uptake the toxic agent or DNA dihexist, they efflux these hex very soon using this pump. Therefore, stem cell population is not stained by DNA dihexist. Therefore, if you isolate the CD31 positive, CD45 negative endothelial cells, so CD31 is the endothelial cell marker, and CD45 is a hematopoietic marker. So we can define the endothelial cells as CD31 positive and CD45 negative cell population. So from hind limb muscle, skeletal muscle, we isolated this endothelial cell population and treated with the DNA dihexist, red or blue. So most of the endothelial cells were labeled by this hexist blue and red here. So these are called main population here. But some of them, so only 1% of endothelial cells are not stained by this DNA dihexist. So they are called side population. So if you treat the endothelial cells with verapamil before the Hextos treatments, you can see this side population disappears because this verapamil is an inhibitor of this ABC transporter. So this is actually the drug-resistant, endothelial cell population. So anyway, we try to isolate this population and this main population and side population and cultured on OP9 stromal layer. So the actually side population endothelial cell generates a very large endothelial cell colony. And if you look at the time-lapse imaging, so we isolated the GFP positive endothelial side population from the skeletal muscles and the cultures. So actually, this cells is a dormant. This cell is the dormant. Therefore, they never divide so quickly. However, after 3 or 4 days of culture, they start to divide and generate a lot of endothelial cells in vitro. Therefore, this side population has such kind of the highly proliferating ability of endothelial cells. But if you culture the main population, so main population barely generate this kind of very small colony. However, the incidence is very low. Only 1 per 100 cell generates this kind of very small endothelial cell colony. So this suggests that the side of population has the endothelial stem cell-like cells. So anyway, we try to isolate this side of population and main population from the GFP mice, and they inoculated into the hind limb ischemia model. So femoral artery as ligated, and then we injected the side population endothelial stem cell population or main population endothelial cells into the hind limb ischemia model. So main population did not induce the recovery from ischemia. However, this side population induced a recovery from ischemia. So this is Doppler analysis. So when the blood flow is observed, red color is detected. So if you observe the phenotype of the blood vessel generated main population, side population, so main population sometimes generate this kind of the cord-like structure. But this cord doesn't contain any erythrocyte in the lumen. So this is a non-functional blood vessel. However, if you inject side population endothelial cells into the mouse, skeletal muscle, so endothelial cell side population driven, endothelial cell generates a whole blood vessels like this. So this endothelial cells, GFP positive endothelial cells and GFP negative donor blood vessels are connected with each other. And then we could detect many, many erythrocytes in the lumen. So this is the actually functional blood vessels. When you observe the histology, so CD31 positive blue endothelial cells were fully covered with SMA positive pericytes, smooth muscle cells around the endothelial cells. So this is the mature blood vessels, probably Planck cells. And from this new blood vessels, another new branch is also detected. So this suggests that the chip cells and stalk cells emerge from this side population. Okay, so this is 3 months after transplantation. But if you observe the 6 months later, so gradually, blood vessels were, replaced by GFP positive endothelial cell like this. So this side population endothelial cells actually generates, sustainable blood vessels in vivo. But if you want to know whether this side population is actually stem cell or not, so stemness should be evaluated by single cell transplantable ability. But in case of the hind limb ischemia models, single cell transplantation is very difficult. Therefore, we change the model from the hind limb ischemia model to liver injury model. So anyway, we try to isolate side population and the main population from the liver again. So actually, we found that side population and the liver actually generates a very large endothelial cell colony on OP9 stromal layer from single cell like this. But main population endothelial cell did not generate such kind of large colony. So this also, as observed in the skeletal muscle, liver also side population endothelial cells is the highly proliferating ability, probably stem cell-like cells. So in order to know whether side population generates new blood vessels in vivo or not, so we utilize the monocrotaline induced liver injury model. So anyway, you can see if you treat the mice with the monocrotaline, sinusoidal blood vessels as very severely damaged. So if you treat some mice with monocrotaline, and then we transplant the side population, main population endothelial cells from the GFP mice into this liver. So you can see, so main population did not contribute to the blood vessels in vivo. However, side population generates a very large sinusoidal blood vessels in vivo. So side population actually, generates a new blood vessels, vessel formation ability. So ultimately, we try to inject a single cell. So if a stem cells, if this is a stem cell, so these single cells can generate blood vessels in the liver. So we isolated the side population endothelial cells from the GFP mouse liver, and these single cells was injected into the neonates, and then observed the liver after mouse grow into the adult. So in the adult liver, you can see single-cell derived vascular unit was developed, continuously induced. And when you observe the, trace lineage by using the GFP positivity in this liver, you can see. So GFP positive cells, most of them are in main population, but some of them are in side population. So this suggests that the side population endothelial cells have the ability to self-renew and generates a lot of the main population. So this is one of the hierarchy from the side population to main population, but this is very shallow hierarchy. Therefore, in order to know the deep hierarchy from the stem cell to terminally differentiated endothelial cells through the endothelial progenitor cells, we need to a very nice marker to identify the stem cell population and the progenitor cell population. So anyway, we compare the gene expression between main population and side population using the microarray analysis. Very fortunately, we identify very nice two marker that is, CD157 and CD200. So those are glycoprotein, which were, reported to be expressed in the part of the B lymphocyte. But so these molecules have not been reported to be expressed in the endothelial cells. So anyway, we try to, observe these two molecules in the liver CD31 positive endothelial cells. So as actually, we could detect the three population, 157 positive and 200 positive cells, and 157 negative, 200 positive, and double negative cells. So we could detect 157 to 200 positive cells are very strongly expressed in the side population. So we thought this population, 6% of all of the endothelial cells may be a stem cell population. And so this stem cell population may be, become to be progenitor cell population after, CD157 disappearance. And probably this population finally differentiated into double negative cells, terminally differentiated endothelial cells. So in order to know that, we initially culture these three populations on OP9 stromal layer. So you can see this 157-200 double positive cell generates a lot of endothelial cell colony from single cells, but 157 negative and 200 positive cells also generates colony. However, the number of the colony is fewer, and the individual colony size is very smaller compared to double positive cells. And double negative cells, as you can see, so cell can survive, but they never show cell division. So they don't have any ability to proliferate, but this double positive cell have ability to generate a lot of endothelial cells. So in order to know this actually stem cell population, we utilize the in vivo transplantation model using the monocrotaline. So anyway, we isolated these three populations, and these populations was injected, transplanted into the mouse liver. So you can see double positive cell generates a lot of sinusoidal blood vessel in the liver. So when you observe the, if you trace the lineage by using GFP positivity, you can see. So this GFP positive anterior cells, so double positive cells expanded, and they finally differentiate into double negative cells through single positive cells. But if you inject this 150 negative 200 positive cells, this green, you can see that the blood vessels is actually observed, but the efficiency is very lower. And if you observe the GFP positive cells in this liver, so these single positive cells give rise to double negative cells, but never give rise to double positive cells. Only this population can generate a double negative cell. And if you inject this double negative cells into the mice, so double negative cell did not generate blood vessels in the liver, they stay as double negative cell. So this suggests that this double positive cell is as apex top of hierarchy of endothelial cells. So ultimately, we also injected only one CD157 positive 200 positive cells into the monocrotaline induced liver injury model. So you can see several months later, you can see one cells generates a very large, vascular area like this. So most of them are in sinusoidal blood vessels, but this population also can contribute to the hepatic vein here, and also the portal vein here. So, and also when you trace the lineage by using GFP positivity, you can see we only inject only one, 157 200 positive cells, but they can expand it in the liver, and they can generate 157 200 negative cells through the 157 negative 200 positive cells. So this is actually the top of hierarchy of endothelial cells. So we can illustrate this kind of the picture, 157 positive 200 positive cells. Actually, this is a stem cell population, and they generate a lot of endothelial cells by producing the, CD157 negative 200 positive cells. So in order to know that these endothelial stem cell population actually exist in the pre-existing blood vessel or not, so we needed to label this endothelial stem cell population by green, and we observe the trace the lineage of this cell population in the liver. So anyway, firstly, when you observe the localization of 157 positive cells in the liver, so these blue colors show the all over endothelial cells here, sinusoidal blood vessels, and this is a portal vein. So only 157 positive endothelial stem cell population localized in the portal vein here, okay? There are no 157 positive cells in the sinusoid. So we label this 157 positive endothelial stem cell population with GFP, and this mice was, induced the liver injury by monocrotaline. So actually, these GFP positive cells started to proliferate on the portal vein, and they started to sprout into the central vein area, sinusoidal blood vessel area, and they generate a lot of endothelial cells in the sinusoid, and then recover the liver injury. But this is too much, so we could not detect the coronary expansion. Therefore, we label these 157 positive cells with GFP with low amounts, and then induce a liver injury. So we actually found that portal vein endothelial cell generates sinusoidal blood vessels. You can see very clearly. So coronary expansion was actually observed in vivo. So anyway, we identify the endothelial stem cell population in vivo. They are marked by these two molecules, and they differentiated double negative cells. This is one of the hierarchy of the endothelial cells. So if you summarize, so we found that endothelial stem cell population in the liver, in the pre-existing blood cells. So I did not show the data, but actually CD157 positive endothelial stem cell can be detected in the artery also, but dominantly, endothelial stem cell population localized in the portal vein, and they start to proliferate and generate endothelial cells upon the vascular injury, and they actually, recover the liver, vascular injury by the differentiation of stem cell to a terminally differentiated endothelial cells. So as I said already before, so endothelial cells has a organ-specific, gene expression cluster character. So we think that the endothelial stem cell population localized in the all of the organ. But we know now so far we don't have any good technique to make a single cell suspension. So, but 3 or 4 years ago, we generated a very nice protocol to generate a single cell suspension of blood vessels in all of organ using the several enzymes. Therefore, using this technique, we isolated the CD157 positive, CD150 negative cell from all of the organ like a retina, brain, heart, skin, muscle, and lung. So as you can see, only 157 positive endothelial cell can generate a very large endothelial cell colony. You can see, but the 157 negative cell did not. So this suggests that tissue organ-specific vascular integrity will be induced by, maybe induced by, endothelial stem cell population localized in the organ. But we don't know whether this organ specificity was observed or not. Therefore, we try to generate a brain ischemia model by occlusion of common carotid artery here. So this is my growth coil. So in this model, 30% of cerebral blood flow is diminished. So in other words, 70% of blood flow is maintained. So actually, but these mice can survive, but they show the ischemia, brain ischemia. So we isolate the brain endothelial stem cell, 157 positive endothelial stem cells, liver endothelial stem cell, fat endothelial stem cell population. So, and then we initially culture these cells in vitro. So the actual liver fat originated 157 positive endothelial stem cell population can generate a lot of endothelial cells in vitro. However, brain C157 positive did not generate so much endothelial cells. However, when you isolate these endothelial stem cell population from the organ and directly inject into the ischemia brain, you can see. So brain-derived endothelial stem cell population generates a very fine capillary network structure in the brain. But the liver fat-originated 157 positive cell did not generate so much in the brain microenvironment. So this suggests that the endothelial stem cell population is organ specific. So the function of the endothelial stem cell population specifically, you induce the brain tissue specific blood vessels in vivo. Okay, so next, second part of my talk is the developments and the aging of endothelial stem cells. So if you observe the liver endothelial cells from the embryo to adult by using the single cell analysis and the flow cytometric analysis, this is by Dr. Fitorian-san in my lab from Indonesia. As she observed the endothelial cell gene expression profile firstly, and the E14.5. This is the primary components, but you can see flow cytometry. We could not detect any CD157-200 positive cell population. So in the early embryogenesis, we could not detect any endothelial stem cell population. However, so this is the same in the 15.5, but the perinatal day before birth, so the 200 positive endothelial progenitor cell population appeared in the mice. But you can see in the liver and P7 after birth, we could detect 157 positive cells, and this 157 positive cell population gradually increase, and then in 14.5, after birth, the number of the endothelial 157 positive stem cell population is equivalent observed in the adult. So I did not show the data, but actually this, 157 positive stem cell population in adult can generate this kind of the adult type endothelial cells by velocity analysis. So this, gene expression is quite different observed in the embryo. So the embryonic endothelial cells and the adult type endothelial cells are completely different genotype. So we recently found that during this perinatal days, so KLF4 and the EGR1, these are transcriptional factors upregulated in the endothelial cells. Probably we think that this population, this transcriptional factor is involved in the reprogramming of endothelial cells to endothelial stem cell population. So why this kind of the endothelial stem cell population developed after birth? So we saw that probably a maturation of liver is induced by endothelial stem cell population, because if you observe the sinusoidal blood vessel from embryo to adult, so in the 15.0 embryonic day, in the early stage of embryogenesis, blood vessels are monotonously dilated, but they don't have any sinusoidal pattern. And P1 is also same. However, in the P7 after birth, you can see central vein and also the portal veins observed. So this is the kind of the hepatic lobe, the appearance. So this expanded and they generates a very complete, hepatic lobules after, 14 and 21 days after, birth. So we think that this kind of the, endothelial stem cell population is involved in the liver donation, because we already know that the portal vein endothelial stem cell population generates this kind of the sinusoidal blood vessels. Therefore, this endothelial stem cell population localized in the portal vein generates sinusoidal blood vessels and induce the liver vascular zonation for the functional liver. Okay, so this kind of the, this is a development of the endothelial cells. So, but in order to know how the aging affects this endothelial stem cell population. So firstly, you imagine how the, vascular density is affected in the several organs and tissues. So this was already published by Dr. Ralph Adams in Germany 10 years ago in Nature. So they found that the humeral head, this is a bone, humeral head suggests that, the vascular density gradually decrease with aging here. Okay, and also another group suggests that in the brain, you can see arterial venous character is not different with aging. However, the capillary density gradually decrease with aging, you can see. So this may be endothelial stem cell population decrease and then capillary density decrease. So we thought that, in order to know that we initially observe the endothelial cells in the liver. So you can see compared to the young adult sinusoidal blood vessels here. So in the 27 months very old mice, the capillary sinusoidal density becomes to be, decreased like this. And also when you observe the CD157 positive 200 positive endothelial stem cell population. So this endothelial stem cell population also decrease in number. So probably endothelium stem cell population will be damaged by the aged microenvironments. Therefore, we try to observe which kind of microenvironment molecular cue affects the endothelial stem cell population. So anyway, we try to isolate endothelial cells from the younger mice and the older mice, and then compare the gene expression between these two endothelial cells by using gene ontology and the KU analysis. So we found that apoptosis and the inflammatory response is upregulated in the older endothelial cells, And also in the KEGG analysis, we found that TNF-alpha signaling pathway is actually upregulated. So as you may know well, the TNF receptor signaling pathway have two different pathways. One is the apoptosis pathway by caspase, and the other is the survival pathway, TAC1, NF-kappa B, and C-flip. So C-flip inhibits the caspase expression. Therefore, this is a survival pathway. So in elder person and elder mice, so probably NF-kappa B pathway upregulated. Therefore, we saw that this pathway may be inhibited. Therefore, the cell death is induced in the mice. Therefore, in order to know that we generate the V adhering free endothelial specific TAC1 knockout. This is TAC1 flux. Therefore, this is the mice was we treated mice with with tamoxifen at 2 to 6 months of age, so older age, and then we observe the vascular phenotype in this younger mice. Again, TAC1 pathway is completely suppressed by using these mice. So you can see, so after treatment with tamoxifen, sinusoidal blood vessels gradually decrease, and then day 10, so this mice died. So severe vascular defect was observed in the TAC1 deficiency. So, but if you observe the day 8, this is quite similar observed in the older mice sinusoid. So probably TAC1 TNF-alpha system is very important for the keeping the survival of endothelial cells and also endothelial stem cell population. So anyway, this is the my a story idea that probably inflammatory signal affects the endothelial stem cell by extrinsically. In this case, that TNF-alpha may damage the endothelial stem cell population and the all the microenvironment, and therefore the decrease of endothelial stem cell population induce the decrease of capillary density. So now we are analyzing how these endothelial stem cell populations are kept in the microenvironment from the niche cell components. So now, as you may know, this is the skin in the 6-year-old human, and this is 75-year-old human. So capillary density gradually decrease with aging, we can see, and also the length of the vasculature is also shortened. So this is maybe involved in the aging phenotype. And also the decrease of the capillary density in the bone may be involved in the osteoporosis. And also the decrease of the capillary density in the brain is associated with the Alzheimer's diseases. Therefore, we should know that how blood vessels is kept in the special endothelial stem cell niche for keeping that blood vessel formation. So the blood vessel formation. Okay, so this is already time. So I would like to say some application of endothelial stem cell population for tissue regeneration therapy, but time is already limited. So I just say that the endothelial stem cell population transplantation is very useful for Heinrich ischemia model. So this is a Berger diseases. So actually the hypoxia is actually recovered from the endothelial stem cell population. And also the endothelial cells not only deliver oxygen nutrient to tissues, they also produce several cytokine or homeostatic factor by angiocrine, as I said in the previous slide. So we now saw that endothelial cells produce the factor 8 expression in the liver, and factor 8 deficiency is observed in hemophilia A patient. But if you treat the hemophilia A model mice with endothelial stem cell population, so as actually endothelial stem cell population generates a blood vessel in the liver, and they secrete many factor 8 in the liver, and so they inhibits the breeding of the mice. So this may be very important very useful for the treatment of the hemophilia A patients. So this is anyway my final slide anyway. So as you may know, well, blood vessel formation consists of the two processes, vasculogenesis and angiogenesis. So mesoderm can give rise to endothelial cells, and they form two blood vessels. But the angiogenesis, the new blood vessel formation, is induced by, from the pre-existing vessels. So now this angiogenesis is actually a very important, involved in the progression of many diseases. But now we know that endothelial stem cell population is very involved in the angiogenesis. So we need to consider about that how endothelial stem cell population is regulated during this angiogenesis, and also the progression of diseases. So, okay, so this work was done by my laboratory, and also I would like to say thanks to Dr. Yoshiaki and Kubota for providing me the VE card free, and Dr. Shizuo Akira for providing us Flox tapeworm mice. So thank you very much for your attention, please. Thank you very much.
Thank you again, Prof. Takakura Sensei. So that those are very important things to consider, and it also gives me some ideas where to go next with the angiogenesis and the liver organoids, Prof. Okay, but this is not about our project. This is about the students who are eagerly waiting to ask questions to you. So now I think I will open the floor for discussion. Anyone wants to pose questions, you can directly open your mic and ask questions. Okay, so I think I will allow participants to unmute themselves. Yes, Dr. Ada has raised her hand. Yes, please, Dr. Ada. And Justin. So I think I would like to introduce Justin as well, Prof. He's a medical student, third year, undergraduate from our Faculty of Medicine, but he has such a tremendous interest in stem cell research. Okay, so I will let Dr. Ada first to give questions, and then Justin. Yeah. Oh, can someone help Dr. Ada?
Okay, yes.
Okay.
Thank
Okay,
you,
thank
Dr.
you.
Nana.
Yeah.
A few moments, I think not allowed to open mic. Okay, thank you, Prof. Takakura. Actually, I'm interested in to elaborate or to explore about the endothelial cell, actually not stem cell, yeah, but endothelial cell of maybe the mice who is the mice which is infected by fungal. What do you think? Because for cryptococcal meningitis, we already know that it's because of the endothelial cell of brain of human could damaged by cryptococcus, and also for mucormycosis, this fungus very progressive, and also there is angio-invasive in mucormycosis person. That's all, Professor. I just curious about it. Is it possible to do in your lab, or maybe you have other idea? Thank you.
So you mean that the infectious diseases is associated with endothelial stem cell population or not? You mean that the endothelial, some fungi infect to endothelial stem cell or not? You asking me? That I cannot catch your question that way.
Can you repeat the question, Dr. Ada? Perhaps simplify or directly go to the question. Hello, Dr. Ada. Okay. Can someone unmute Dr. Ada? Nova?
Yes.
Okay.
Thank you, Dr. Nana. Thank you, Mbak Nova. Okay, I will repeat, Prof. Takakura. I think not really in stem cell, but actually I want to explore what happened in the endothelial cell of mice with cryptococcosis and mucormycosis, because in these two fungal diseases, there is angioinvasive of these fungi to vesicular apa ya, Dr. Nana, to
Yes, so it's kind of like histopathological invasiveness from the two fungal infections
yes,
to the
angioinvasive,
endothelial cells in the
yes,
mice.
to
And
endothelial cell. Yeah.
okay, and you wanted to know whether you can use the tools that has been not provided, but according to the publication in Professor Takakura Sensei lab, uh, for your own research. Is that it, Dr. Ada? Is there any tools, Prof, that she can use to monitor or to evaluate the progression of the fungal infection in the mouse model, Prof? And which mouse model angiogenesis assay, in vivo assay that you can recommend? I think, yeah.
Yes, thank you.
I thank you very much for your question, but I cannot imagine what kind of the model is adequate for seeing that this kind of the infectious disease and also the endothelial angiogenesis so far. So as far as I know, we don't know such a adequate model at this moment. Sorry. Yeah.
Okay, thank you, Dr. Ada. So I think I will go to the next question. Justin? Yes? Can you open your mic?
Okay, thank you, Doctor, and thank you, Professor, for the very insightful lecture. Perhaps I have two questions. The first one is, has there been any research done in the orthopedic application of endothelial stem cells? For example, in aging disorders such as when people undergo osteoporosis, I think that the application of angiogenic stem cells could help to combat that disorder. And the second question is, has there been any reported side effects of possibly carcinogenic nature of endothelial stem cells due to other angiogenic drugs that could possibly cause cancer due to the rapid proliferation? Perhaps those are my two questions. Thank you, Prof.
Second question is about the cancer developments by endothelial stem cell. But so now we are analyzing the 157 positive endothelial stem cell population in cancer is very high in number. And they actually generate a lot of endothelial cells in tumor microenvironment. And they actually induce the immature blood vessels in the tumor. So this is very, important. So we needed to mature the endothelial cells in the tumor microenvironment to, induce a, drug delivery into the cancer microenvironment. So this is so we utilize the CD157 positive endothelium stem cell population for the drug delivery into the cancer microenvironment. So in the first question is the aging with endothelial stem cell. So so what that today I showed that in the brain ischemia models, as actually the dementia was observed in the mice after occlusion of the carotid artery, so common carotid artery. So this endothelial stem cell, if you transplant the endothelial stem cell population into the mouse ischemia model, so dementia is actually improved. So actually the endothelial stem cell population transplantation is useful for such aging, related to diseases actually. Thank you very much.
Thank you very much. Thank you for your question, Justin. So we have another question from Dr. Arum Naruni. Dr. Arum Naruni, would you like to open mic? Dr. Arum? So he posted the question in the chat room. Dr.
Yes.
Arum, are you still here? Okay, so he asked about, can we use CD157 marker to isolate endothelial stem cells, or are there any other markers that should be added or used in the isolation process?
So you can utilize the CD157 as a marker. So we have several antibody, commercially available 157 body antibody. So you can easily isolate the 157 positive if you make a single cell suspension in several organs. So, but in the peripheral blood, we do not have any CD157 positive endothelial stem cell population. But so in this case, we need to make a single cell suspension from organ to isolate the endothelial stem cell population by using the 157 positive antibody. And of course, another marker may be available. Another group may suggest that endothelial protein C receptor, EPCR, is also the marker for endothelial stem cell population. So the EPCR is also available to commercially available antibody. So CD157 or EPCR could be used for isolation of endothelial stem cell population. Thank you.
Thank you, Dr. Arum. I see Dr. Dewi has raised her hand. Yes, Dr. Dewi, can you ask the question?
It's mute. Okay, okay. Yeah. Thank you, Dr. Rana. Thank you, Professor Takakura, for a nice speech. I have two questions actually regarding the endothelial stem cell. First is actually what is the originality of this cell? I mean, for EPC or endothelial progenitor cell, it is stated from the bone marrow. How about this cell? And the
Yes.
second, within the mature endothelial, could you explain the exact or probably the location of this endothelial stem cell? Because theoretically, blood vessel actually is lined up by one layer of endothelial. So can you explain where is the exact location for this endothelial stem cell?
Okay.
Thank you very much.
So many people ask me that what is difference of the 157 positive cells and the EPC in the bone marrow. So we could not detect any 157 positive cells in the bone marrow. So bone marrow endothelial progenitor cell population may express CXCR4 chemokine receptor, but CD157 positive cells do not express any CXCR4. So I think that and also if you generates the bone marrow transplantation model in the mice. So we could not detect any, bone marrow derived 157 positive cells. So maybe the origin of the EPC and the CD157 positive endothelial cells are different. So originally different. So anyway, we could not compare these two cell population, but the EPC may be localized in the bone marrow. But the EPC is the endothelial stem cell population localized in the blood itself. So I did not show so clear data, but in the liver, it's actually portal vein endothelial cells contain a stem cell population. So this cell population is incorporated in the surface of the inner surface of the blood vessels. And also I did not show the data, but so artery aorta also have endothelial stem cell population that is located in the inner surface of the blood vessels. So they do not have any specific localization, but anyway, they act as endothelial cells. Endothelial stem cell work as endothelial cells, a part of endothelial cells in adult. Thank you very much.
May I clarify or confirm? So you mean that this endothelial stem cell is kind of integrate with the mature endothelial cell?
Yes, that's right.
Okay, thank you very much.
Thank you, Dr. Dewi. So I would also like to invite another one. This is from the National Research Agency, Prof. So in Indonesia, we have BRIN. These are pure researcher and a very bright person, Dr. Elra Derovani, my collaborators. Hi. Okay, you can ask questions.
Ya, thank you very much, Dr. Radiana. Selamat sore. Selamat sore, Prof. Takakura. Thank you very much for your presentation. It's very excellent. I'm interested in, especially, in fact, I just have a brief request if you would like to. This is my request. I mean, would you like just share the angiogenesis in during embryo development, and the second, what's the significant differences during development between the human and animal angiogenesis? Thank you, Prof.
Oh, okay. So as I said, in at least in the liver, we don't have any stem cell population during early embryogenesis. So probably as yolk sac blood vessels develop as a primary vascular plexus, so endothelial cells in the embryo is completely different from adult type endothelial cells. So I think that from such kind of the immature embryonic endothelial cells gradually, deprogrammed into the adult type endothelial cell. So that the second question is similar, but anyway, if you generate the endothelial cell from IPS, human IPS cells, so initially we could not detect any 157 positive cells among the early development of the endothelial cells. But after generation of many endothelial cells, among that we could detect 157 positive endothelial cell population. And if you compare the CD157 positive and negative endothelial cells from the human IPS, so only 157 positive endothelial cell can generate the vascular network in vitro and in vivo also. This may be 157 positive endothelial stem cell population develop later stage of embryogenesis, and then they can contribute to the adult blood vessels after birth. I think so. That is
Thank you very much.
now submitting this paper at this moment. So.
Makasih,
Thank you so
Dr.
much.
Diana. Yeah.
Thank you so much, Takakura Sensei. So because of the time, again, I think we need to conclude this wonderful guest lecture and very insightful, very meaningful and purposeful guest lecture about angiogenesis and vascular endothelial stem cells. I'm sure everyone here learned so much from you. And as we mentioned before, this will be kept in the digital archive of Doctoral Program Biomedical Science and Master Program Biomedical Science. So each of our students, all of our lecturers can access this archive and learn more from your presentation, as well as search for the publications and cite the publications as well. Thank you so much, Takakura Sensei. I would also like to thank our international guests here, Dr. Bal Zainab, Dr. Fitriana Nur Rahmawati, and saya Nuralda Sitampu. Thank you so much for attending the guest lecture, as well as lecturers in Faculty of Medicine, Universitas Indonesia, which I see here. We have Dr. Adisti Dwi Jayanti, Head of Department of Pharmacy, Ms. Dr. Arleni Bustami, also a cell culture expert in our Faculty of Medicine. So thank you so much, everyone, for joining. I think I will hand it over to the Master of Ceremony, Nuzli Fahdia Maskufa, for closing and memento. Thank you so much.
Okay, Thank you for the amazing presentation and lecture, Professor Nobuyuki Takakura. Actually, I have a few questions, but we are limited in the time. So maybe after this, I will have a conversation again with the Dr. Radiana because this topic is very interesting, right? So I hope all participants also will get the new understanding about the concept and the development, also the clinical potential about the angiogenesis and vascular endothelial stem cell. So everyone, we have to come to the end of today's event. But before we close this event, we will hand over the certificate as a memento for the Professor Nobuyuki Takakura. And I will share screen. Wait. Yeah, okay. For the Professor Nobuyuki Takakura, there is a memento for us, and I give over to the Dr. Radiana again. Doc?
Okay, Dr. Ada, would you also like to say something? I mean, Prof. Takakura Sensei, we hope that this will not be the last time you share your valuable science and technology to our undergraduate medical students, master students, and doctoral students in Faculty of Medicine. We hope that this will continue onwards and that if you come by to Universitas Indonesia next year, you can give a direct lecture to all our students. So thank you so much for today, Prof.
Yeah.
Okay.
Oh, okay, before we ended the session, maybe we can took the photo together for the attendance. Maybe you can open your camera and then we can take the screenshot for the photo. Okay, so everyone, please open the camera. Okay, so we have 2 pages in here, still waiting. Okay. Okay, I think it's enough. Okay, we took the picture. Everyone's ready? I will count in 1, 2, 3. Okay. The next pages. 1, 2, 3. Okay, once again. Okay, yeah. I think that's enough for the today's session. So we hope all the participants get the new experience and understanding about the angiogenesis and vascular endothelial stem cells. Thank you so much for today's participant and especially for the Professor Nobuyuki Takakura. We hope we can have other lectures from you here in the Faculty of Medicine of Universitas Indonesia. And also we thank to our skillfully moderator today, Dr. Adiana Dewayani Antarianto. And I think I will close this today's event right now. See you on the next event. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you, Sensei.
Thank you very much.
Thank
See
you
you.
so much.
You.
Okay. Okay, thank you. Bye-bye. So we will leave now. Thank you.